Alright, so now we're going to talk about viral vaccines. Vaccines against viruses play an important role in protecting humans against infection from viruses. There are three categories of viral vaccines: live attenuated, killed, and recombinant. They are listed here for your reference. Let's start with live vaccines. Live vaccines incorporate attenuated or weakened viral strains and are effective against both naked and enveloped viruses that require cell-mediated immune responses to clear. The response to a live virus will be Th1 and Th2, thereby stimulating longer-lasting immunity through the development of humoral, cellular, and memory immunity. If none of that makes any sense to you, and you need a refresher on the different types of immunity, go right now and take a look at the immunology section. The risk with live vaccines is that they can revert to active virulent strains. I've listed here for you the different live viral vaccines. They include smallpox, yellow fever, varicella vaccine, the Sabin polio vaccine, MMR vaccine, and the intranasal influenza vaccine. Let's look at inactivated vaccines. Inactivated vaccines contain killed virus with immunogenic stimulants such as the viral capsid or envelope antigens. The immune response is predominantly Th2, thus generating only a humoral or antibody response with no strong cell-mediated response. Therefore, the immunity is short-lived and requires boosters to maintain. However, inactivated vaccines are safer, especially for immunocompromised patients. The list of killed or inactivated viral vaccines includes rabies, intramuscular influenza as opposed to the intranasal influenza, the Salk polio vaccine as opposed to the Sabin vaccine, and hepatitis A vaccine. One great question that they love to ask on the step 1 is which vaccine you can give to an HIV or otherwise immunocompromised patient. You can definitely give killed vaccines. Remember that the MMR vaccine is the only live vaccine that you can give to an immunocompromised patient. You may be wondering, why is this the case? Well, remember that it is a balance between the immunocompromised patient potentially contracting the viral infection from the vaccine versus not being protected from a potentially fatal disease. In the case of the MMR vaccine, it was decided that the benefits of the vaccine far outweigh the low possibility of seroconverting from the vaccine itself. Let's talk about the last category of viral vaccines, recombinant vaccines. So, what exactly is a recombinant vaccine? It requires the use of a vector. A vector refers to the use of a carrier bacterium or virus. Essentially, scientists have created recombinant vaccines using genomes of either harmless or attenuated viruses, with pieces of genetic material from other viruses that have been inserted through recombination, the process that we discussed earlier. The vector or carrier virus can then take this DNA to the host cells. In other words, because this mimics a natural infection, it will be able to stimulate the immune system. There are two recombinant vaccines that you should know about, and those are the HBV and the HPV vaccines. Because they sound so similar, they should be easy to remember. Alright, let's do a flash quiz. Here's a true-false question. You can give the MMR vaccine to an HIV-positive patient. This is true. Remember, this is the only live vaccine you can give to an immunocompromised patient. The reason the vaccine is recommended is because of the measles component in the MMR mnemonic. There are a significant number of measles cases in the USA, and the mortality rate from measles in these patients is extremely high. Thus, it's prudent to vaccinate these patients. Okay, so as I mentioned before, learning the characteristics of viruses is painful. You must be really wringing your hands right now and shaking your head at me as I carry on and on about these different characteristics of viruses. However, I will point out that this can actually be high yield, especially when a step 1 question gives you an easy to identify virus, but then forces you to answer the question by choosing the answer that identifies the virus. So my advice to you is to remember the general rules and the few exceptions. If I were you, I would come back to this point of the section when you have completed the rest of the virology section. It will be a good way to tie in all the information that you have learned. So, here's how I remember the basic genome information. Remember that all DNA viruses contain double-stranded DNA, except for parvovirus, which has single-stranded DNA. It is helpful to remember that this is the mnemonic, that all are double-stranded DNA except for parvovirus, aka parvovirus, which is single-stranded. Also, try to remember that all are linear except for these three, namely papilloma, polyoma, and hepadna. Let's move on to the RNA viral genomes. In contrast to the DNA viruses, remember that the RNA viruses almost all contain single-stranded RNA. The exception to this rule is the reoviruses, which are double-stranded. The most important member of the reovirus family is rotavirus, which causes diarrhea, commonly in infants. When we talk about RNA viruses, we can differentiate between positive and negative-stranded RNA viruses. Positive-sense RNA means that it has the same polarity as mRNA and can therefore be directly translated into viral proteins without any further processing. In contrast, negative-sense RNA is complementary to viral mRNA, and therefore it needs to be converted to positive-sense RNA by an RNA polymerase before it can be translated. After you become more familiar with the RNA viruses, which we will discuss soon, perhaps this mnemonic will help you to remember which ones are the positive-stranded RNA viruses. I went to a retro toga party where I drank flavored Corona and ate hippie California pickles. Alright, flash quiz again. So, help me fill in the blanks. DNA viruses are usually blank-stranded, while RNA viruses are usually blank-stranded. Easy enough, right? DNA viruses are usually double-stranded, while the RNA viruses are usually single-stranded. Remember the exceptions. This next point highlights an important question. Are the purified genomes of naked viruses infectious? Well, let's talk about it. It actually depends on the type of virus. Most double-stranded DNA viruses are infectious. For RNA viruses, purified nucleic acid from positive single-stranded viruses are also infectious. On the other hand, purified nucleic acid from negative single-stranded RNA and double-stranded RNA viruses are not infectious on their own. Now, do you understand why this is the case? This looks like a very random list, doesn't it? Double-stranded DNA does not have anything outside of its genome that it needs to be able to enter a cell, replicate, and therefore be infectious. The two exceptions that you need to know are poxvirus and hepatitis B virus because they require their own polymerase, which they carry with them in the virion. Similarly, as we already discussed, single-stranded positive-sense RNA is already like mRNA and is ready for translation and therefore can be infectious on its own. However, it is the negative-sense single-stranded RNA which needs the RNA-dependent RNA polymerase to be converted to its complementary strand before it can be translated. The same is true for double-stranded RNA viruses. This is why negative single-stranded RNA and double-stranded RNA viruses are not infectious when they are naked. Okay, so now we're going to talk about viral replication. I'm sure a very interesting topic for all of you. Let's start with this image. We're going to talk about DNA viruses as compared to RNA viruses. This is an image of a DNA virus. It's a visual representation to help you remember how viruses replicate. The rule is that DNA viruses replicate in the nucleus. This means that they enter the nucleus and use the host cell's DNA-dependent RNA polymerase to synthesize mRNA from its negative-stranded template. The exception is poxvirus, which has its own DNA-dependent RNA polymerase and therefore does not need to replicate in the nucleus using host cell polymerase. Hepatitis B virus is another unique virus because its double-stranded genome contains single-stranded regions scattered throughout. We'll talk more about its replication soon, but it actually starts part of its replication in the cytoplasm before completing it in the nucleus. Now, for RNA viruses, the rule is exactly the opposite. Here's another image to help you remember this rule. For RNA viruses, the rule is that they replicate in the cytoplasm. The exceptions are listed here. They are influenza and retrovirus. We're going to discuss these viruses in more detail when we get to them. Alright, so we've already discussed what a naked or non-enveloped virus is compared to an enveloped virus. So do you remember what the envelope is? Here's my squiggly representation to help you remember what the envelope is. You have the genome surrounded by the capsid, and outside there may be an envelope. The envelope is the additional lipoprotein bilayer around the capsid. You might think that the envelope provides an extra protective layer, but this is not true. In fact, enveloped viruses are highly susceptible to damage by conditions such as detergents, extreme pH, temperature, and organic solvents such as ether. On the other hand, naked viruses are usually more stable than enveloped viruses because they do not have a lipid-containing envelope and can therefore withstand these agents. So I've written it out for you. The enveloped viruses are weaker than if they were naked. So how do enveloped viruses get their envelopes? As you can see in this image, enveloped viruses acquire the envelopes from the host cell's plasma membranes when the virus exits the host cell. The exception to this rule is with herpes viruses, which actually get their envelope from the inner nuclear membrane. There are 8 naked viruses. 4 are DNA viruses: papillomavirus, adenovirus, parvovirus, and polyomavirus. To help you remember this, think of a pap smear. The other 4 naked viruses are RNA viruses: calicivirus, picornavirus, reovirus, and Hepi. To help you remember this, think of the mnemonic CPR plus H. Alright, flash quiz again. Let's drill this in. Most DNA viruses replicate in the blank, while most RNA viruses replicate exactly where? Right, you know this. Most DNA viruses replicate in the nucleus, while most RNA viruses replicate in the cytoplasm. So now let's just quickly summarize the DNA virus characteristics we've already learned. You'll have seen part of this table before. We're just adding a few things on. My recommendation is that you do not spend too much time learning these, as it is not high yield. Again, focus on big picture characteristics that several viruses share, and also know the exceptions. DNA viruses are double-stranded, linear, icosahedral, and replicate in the nucleus. These are the exceptions. The DNA viruses are very happy viruses and include hepadna, herpes virus, adeno, pox, parvo, papilloma, and polyoma viruses.